Antigenic variation allows pathogens to escape from immunity. A second, more dynamic mechanism of antigenic variation is seen in the influenza virus. At any one time, a single virus type is responsible for most infections throughout the world. The human population gradually develops protective immunity to this virus type, chiefly by directing neutralizing antibody against the major surface protein of the influenza virus, its hemagglutinin. Because the virus is rapidly cleared from individual hosts, its survival depends on having a large pool of unprotected individuals among whom it spreads very readily. The virus might therefore be in danger of running out of potential hosts if it had not evolved two distinct ways of changing its antigenic type. The first of these, antigenic drift, is caused by point mutations in the genes encoding hemagglutinin and a second surface protein, neuraminidase. Every 2 to 3 years, a variant arises with mutations that allow the virus to evade neutralization by antibodies in the population. Other mutations affect epitopes that are recognized by T cells and, in particular, CD8 T cells, so that cells infected with the mutant virus also escape destruction. Individuals who were previously infected with, and hence are immune to, the old variant are thus susceptible to the new variant. This causes an epidemic that is relatively mild because there is still some cross-reaction with antibodies and T cells produced against the previous variant of the virus, and therefore most of the population have some level of immunity. Major influenza pandemics resulting in widespread and often fatal disease occur as a result of the second process, which is termed antigenic shift. This happens when there is reassortment of the segmented RNA genome of the influenza virus and related animal influenza viruses in an animal host, leading to major changes in the hemagglutinin protein on the viral surface. The resulting virus is recognized poorly, if at all, by antibodies and by T cells directed against the previous variant, so that most people are highly susceptible to the new virus, and severe infections result. The third mechanism of antigenic variation involves programmed rearrangements in the DNA of the pathogen. The most striking example occurs in African trypanosomes, where changes in the major surface antigen occur repeatedly within a single infected host. Trypanosomes are insect-borne protozoa that replicate in the extracellular tissue spaces of the body and cause sleeping sickness in humans. The trypanosome is coated with a single type of glycoprotein, the variant-specific glycoprotein, VSG, which elicits a potent protective antibody response that rapidly clears most of the parasites. The trypanosome genome, however, contains about 1,000 VSG genes, each encoding a protein with distinct antigenic properties. Only one of these expressed at one time by being placed into an active expression site in the genome. The VSG gene expressed can be changed by gene rearrangement that places a new VSG gene into the expression site. So, by having their own system of gene rearrangement that can change the VSG protein produced, trypanosomes keep one step ahead of the immune system, capable of generating many distinct antibodies by gene rearrangement. And that's antigenic variation allows pathogens to escape from immunity. Have a great day.